Hi everyone, good afternoon. I'm Amrutha Varshini.
And I'm Sinead Robinson-Cast.
So today we are presenting a paper on
Protein sequences from Mastodon and Tyrannosaurus Rex revealed by mass spectrometry.
Yeah, that's right. So on to our slides.
Yeah. The authors of this paper are Asara, Schweitzer, Freimark, Phillips, and Cantley from Harvard Medical School, North Carolina State University, and Beth Israel Deaconess Medical Center. The paper was published in the journal Science in 2007. Science is an incredibly influential journal. It has an impact factor of 41.04 as of 2018, and this particular paper has been cited 256 times since publication.
Fossils from extinct organisms provide the opportunity to reveal evolutionary links between extinct and modern extant animals. For this paper, the focus was on 162,600-year-old Mastodon and a 68 million-year-old T-Rex. The Mastodon sample was a long bone with a soft tissue preserved from a specimen in the Museum of Rockies. The 68 million-year-old T-Rex sample was a femur found at the base of Hell Creek Formation, also with preserved soft tissue. Collagens are the most abundant protein found in bone, greater than 90%, and they have specific PTMs. Their longevity has been proven in other papers, and they're also highly conserved, making them the perfect candidate to sequence. The collagens looked at this paper are mainly A1T1 and A1T2. As stated, sequencing samples from such ancient specimens means facing a number of challenges with regards to acquiring intact samples and without access to a sequenced genome. The advent of mass spectrometry allows us to overcome these issues. Mass spectrometry technology has allowed us to sequence fragmented proteins and peptides in quantities as small as sub-picomolar amounts. MS software generally relies on exact sequence matches, so unless they match exactly, they won't be identified. To get around this challenge, the author used a two-step proteomics approach using ion trap mass spec fragmentation patterns. After sequencing the collagen samples from the bones, they looked at tryptic peptide fragments that matched identically to orthologous proteins or proteins from extant taxa. Next, they generated a database of likely drifts in amino acids by comparing the sequences of the orthologs from multiple related taxa. This created a bundle of theoretical protein sequences, which were then compared to the fragmentation patterns of peptides taken from extracts of fossilized bones that then match any peptides in the public database.
In order to prove this concept actually works, they took collagen from the femur bone of an ostrich. Using the sequest algorithm, they found 87 tryptic peptide spectra by microcapillary LC-MS-MS that were matches to related organisms in the protein databases. It matched chicken collagen A1T1 the most closely. About 33% of sequenced collagen A1T1, 16% of A2T1 were identified in the ostrich bones through identical matches to specimens sequenced in a public database. One issue that is raised is the potential for peptide sequences to be missed due to evolutionary divergence of amino acid residues in the current database. To address this, the team generated a sequence for the most closely related sequence, the chicken, and compared it to the next most closely related, the frog and the Japanese newt. Where there was divergence across the three amino acids, a theoretical sequence was generated to include the exact sequence in regions where all of the three specimens were identical. They also generated various combinations of the variant residues where the species diverged, working on the assumption that the drift from chicken to ostrich was most likely to occur at the same divergence as in frog or newt. This allowed for a theoretical drift to be predicted and matched MS-MS fragmentation patterns from ostrich bone and the synthetic peptide. There were also more synthetic sequences created for the misaligned residues using PAM matrices. This approach found 6 more matches and increased the coverage from 33 to around 39%, and shows the differences between ostrich and chicken and the potential evolutionary divergence. Applying this technique to the Mastodon sequences found 74 tryptic peptide MS-MS spectra that matched extant mammals in the database. This is 32% coverage for A1T1 and 29% for A2T1. Using MS also allowed them to see that the hydroxylation of glycine was around 50% higher than it had been in ostrich, whilst the proline and lysine hydroxylation were very similar. The Mastodon sequences were found to be most closely related to dog, bovine, human, and elephant than to non-mammalian taxa. The same approach as used in ostrich was then used to identify unique mastodon sequences. Drifts in human, dog, and mouse were compared, and PAM was used to create additional theoretical sequences. 4 extra peptide sequences were found from fragmentation patterns. These additional sequences increased coverage of A1T1 from 32 to 37%. This is a similar increase as seen in the ostrich. This means that they managed to obtain as much coverage of collagen from a recent ostrich bone sample as for a potentially 600,000-year-old fossil.
In specimens of this age, generally there is a lot of degradation, so the tryptic peptides were purified multiple times through solid phase extraction, strong cation exchange, and reversed phase microchromatography. This was done to get rid of a rust-colored contaminant and also to increase concentrations of the peptides. The MSMS spectra obtained showed 7 collagen peptides that could be aligned with predicted fragmentation patterns from extant taxa in the public database. MS showed that most of the proline, lysine, and glycine residues were hydroxylated. A blast alignment was done to search for similarities between the collagen against those in the database. 58% sequence identity was found with chicken, 51 to frog, and 51 to newt. To validate this data, they presented one of the experimental T-Rex sequences that had matched with chicken and the synthetic peptide. The graphic shows that the experimental peptide, the T-Rex, on the left, has more noise and is a low intensity, not surprising given the age of the proteins. No unique T-Rex sequences were found.
Oh, right. General critique. So overall it was a good paper. It was a lot of theorizing though. I think it has new possibilities for mass spec technology. So other people have done further studies. There have been confirmations that the Mastodon sequences were correct, but as far as I can find, there haven't been any confirmations of the T-Rex sequences. In fact, one study actually found that the sample was contaminated.
Papers like these are extremely important to know the evolutionary relationship between ancient and modern species and the involvement of recent technologies like mass spec in finding what was never found before.